All right, so let's start 801. Probably one of the most anticipated lecture topic. A lot of slides, like 50, 50, 55 or 56 slides. But as I said, some of the material is very descriptive, and you can just read the slide and you will understand that. So I will not spend much time on where it is just descriptive information. I will spend more time on the ones where you need to understand the technique, that how the vaccine is developed and what is the mechanism. Those kind of mechanistic questions are answered in some of the slides, and I will just stop in those slides a little bit more. All right. So here, this is 1900 to 2011 life expectancy in the United States. And very interesting part is that this is just not, if you see that how the life expectancy is increasing, say around from 47 to 80 plus, this is not entirely due to the vaccination. Remember that. Along with that, there is antibiotics as well. But medicine basically increase the lifespan of the human being. That's bottom line. And vaccines and antivirals, along with the antibiotics, played an important role to it. So here you can see that this dip is post-World War I. But there is no dip as such after the World War II, maybe a little bit here. But antibiotics were already discovered by 1923, came to the market in around 28, 30, and they were available. So you can see that overall that they are proven best against the viruses with the vaccines. And these are the things that we already talked about in our adaptive memory and the innate memory. That first you get the infection, you get an initial immune response, and then if you get a reinfection as well, you call the immunological memory, because some of the T cells and the B cells which see the infection in the first time, or the antigens, they just remain in your body for the rest of your life. Now, this level may go up or down, but some of these T cells or B cells, clonal, remain. And the second time, you should not apparently get the information. Now, what happens in many of the viral infections, constantly you are exposed to different set of viruses, and your immune memory cannot keep up. Common cold, very common. As such. Some you have to take vaccines pretty regularly. Some you're protected for life. If you take the polio as a child, you're protected for the life. So it just depends. All right, so first, before we go to the proper vaccines, that immunization is a term that we use where we use the immune products, and vaccine is also an immune product, but there are two types. One is an active, which is the long-term protection. That is basically the vaccine that we talked about. Whereas the passive immunization is a short-term protection. We do not call this vaccination. This is more of a therapeutic purpose, and what you use is usually you can use a convalescent plasma which contain the antibodies from another patient who actually saw that, or monoclonal antibody treatment that we talked about in the special topic. So here what happened, that this is also the natural passive vaccination from mother to infant in per se, but which is the passive immunization. So you can easily see that the IgG, or the immunoglobulin G, actually can cross the placenta. That's the only one. And then you can see that over the period of time, the mother's antibody basically decline. But usually what happens, that still after birth, there are IgAs and the IgGs that can be delivered from the breast milk from the mother to the child as well. So these are the ways that the child is protected. But you can see that approximately about 6 months or so, gradually the different IgGs of the child actually goes up, and by the age of probably 5, 6, 7, at that time, you have the normal level of the IgG, IgM, and the IgA. But usually, most of the vaccines are given after 6 months. This is the kind of like a bottom line, and they just allow the mother's antibody to decline, because what can happen, that many of the mothers, this is an interesting part, that many of the mother's antibody can be against the antigen that the child is going to see as well. So what's going to happen that, for an example, mom has a polio vaccine and she does still have antibodies against the polio. Now the child got the vaccine, and before there are still mom's antibody which can recognize them and neutralize them. So you may not get a proper immune response, so they just wait until the passively transferred maternal IgG kind of goes down. So that's one of the rationale for that. Then what we are looking at is that this is a therapeutic antibody therapy, mainly monoclonal antibody therapy, and it has just gone into a state which is completely mind-boggling. So what people used to do is like mouse antibody against the viral epitope, and then you isolate the mouse cells, you make a hybridoma, which is a fused mouse B cell with an immortal B cell, and then that will secrete these antibodies specific to the antigen, and you just purify those monoclonal antibody from just one B cell which is producing that and inject to the human being. Now, problem is that if it's a mouse antibody, which was traditionally used for a long time, the other part, say that right here, you have the antibody part, you already know that you have a light chain and you have a heavy chain. This is the recognition site, which is the variable heavy, variable light, variable heavy, variable light. And then you have a constant region, which is similar in each and every IgG or the isotype. Now, what happens in that these are mainly IgGs. The other part of the antibody, this is foreign to the human being. So when you inject a mouse antibody, your own immune system is going to attack these part and neutralize the antibody itself, and the efficacy of the antibody will go down. So what you do is like now you have a human antibody against the virus or the pathogen, and here you will not have any immunogenicity. What now people do is like, this is an extremely complex genetic manipulation, is graft the mouse viral antigen recognition site from here onto a scaffold of a human antibody, and you have a chimeric antibody. And again, the chimeric is a very empirical term. There is very different chimeric antibodies. We're going to see that. And not going into the detail, you can read that how they are being generated. It is beyond the scope of this class, but I have just put it in the narrative that how it is done. So these are all four different types of monoclonal antibodies that can be given. So these are produced in the mouse hybridoma. So this is original mouse. This is chimeric, where the variable parts are replaced. Here, the humanized part is only the yellow part that is recognizes the antigen that is from the mouse. Every amino acid sequence in between them, which is a scaffold, is from the human being as well. So there is literally no part which is not human in this one. And now what you have, this is the Frankenstein mouse, where the mouse are making the human antibody. This is a transgenic development that is unbelievable, that what you do is like you delete all the B cell genes in the germline of the mouse, and you transfer all the B cell genes that you have, or not the B cell genes, I'm just take that word back, the antibody producing genes, the variable light, heavy, all those, transplant those genes into the transgenic mice, male as well as female, and their offsprings are going to make human antibodies in the mouse. So it is a kind of a mind-boggling development. I have put the procedure that how you basically do that. So these are from the hybridoma. This is the mouse itself making from the transgenic mouse with the human hybridomas. So it's even, it is really mind-boggling that what you do. So basically this is the CDR grafting, is that these areas are called CDRs, which are called the complementary determining region. These yellow parts are the ones, these amino acid sequence basically complement to the antigen. And that's what you are basically doing. So why you are getting these mouse antibodies? Because see that otherwise you have to get these from a human being who is infected with the virus. Here, you just inject the antigen or the virus to the mouse to raise these. And now if the mouse makes the human antibody, you inject the antigen to the mouse, and this will be a human antibody that will be raised in a B cell, and you can just, they will secrete that and you can purify that. So really amazing technology. This just gives you an other idea that how a mouse antibody is more immunogenic and now humanized, and then even human antibodies produced in the mouse, they are completely, there is no immunogenicity as such, because the human body basically don't think this as a foreign protein. Even in there are some of the humanized part, they may think, but here there is no cross-reactivity of the antigen. So this is basically the monoclonal antibody treatment, passive immunization, completely therapeutic purpose. This does not solve the bigger problem because this is very expensive and only you can just do these therapies in extreme conditions. So this is also another example of the Z-map of the Ebola virus, and saved 3 US personnel a couple of years back. So this is just a scaffold where you are seeing that this is the membrane glycoprotein right here, the receptor binding site on like a model on an envelope of the Ebola virus. And these are 3 monoclonal antibodies, just like a model, and just only the part which recognizes 3 epitopes on the surface glycoprotein of the Ebola and can neutralize that. So even after getting the virus, they can be neutralized. So think about that. They are produced in tobacco plant, recombinant tobacco plant. These antibodies, this is kind of amazing. All right, so now let's get into the vaccine, real vaccine. So first, what you have, several different types we are going to talk about. That attenuated vaccine, these are the live viral vaccine, attenuated virus, which doesn't cause the disease. Then you have a recombinant vaccine, which is a DNA mRNA vaccine. We are going to talk about that. Inactivated vaccine, dead or fractionated virus. And then subunit vaccine, which are the protein, peptides, or the nucleic acid of the intact virus. On the other hand, there are other couple of vaccines like a toxoid, tetanus toxoid, carbohydrate vaccine, some of the conjugated protein carbohydrate conjugates. We're not going to discuss these because these are mainly implicated in bacterial infection. These are mainly bacterial vaccines, but these are the vaccines that are against the viral infections. So let's take a look at this. So this is what I just wanted to you guys to just skip over. You can just talk about these Jenner, where he observed that the milkmaids do not get these cowpox blisters in their hand, but they're immune to the smallpox. And Jenner vaccinated James Phipps, and that was all the start with that. And you can just read that, so a very straightforward idea. So you can see this. I'm sorry for the picture, but the smallpox, they used to take this bifurcated needle and just put it on the skin where it used to put this lesion, primary vaccination site, but it protected and basically eradicated the disease from the planet. This is the only disease that completely eradicated from the world. You don't get the vaccine anymore. Polio, almost at the verge, but still there are some pockets in Afghanistan and Pakistan where you still have the polio. And unfortunately, we're going to talk about that. And those are the things that I'm going to discuss in here, that why there is still a fear of vaccine, that what you see here, even after 2 centuries, because there are some reason for that. So that's the reason that what you guys can do, that again, do not just preach people that you need to take a vaccine. If they're confused, just tell them the science behind that vaccine. That will make them understand that what it is. Say, for example, we're going to talk about the polio vaccines and why there is a fear and where there shouldn't be any fear. So that's what I'm going to just talk about that, completely from a scientific point of view. All right. So here, these are some of the other vaccines: rabies, yellow fever, influenza, polio. Now, Jonas Salk, that was the first one, which is an inactivated vaccine, injected 4 shots, inactivated, which could not actually eradicate polio, but almost. Then the Sabin's vaccine, Albert Sabin, this is oral, 4-dose, attenuated 3 strains of IVV. So think about that you take 4 doses. So some of the vaccines needs multiple doses. So when you're just trying to get a booster shot for the SARS-CoV-2, there is nothing to think about that why we need. You get 4, even for polio, which is an attenuated live vaccine. So we're going to see that. Interestingly, what happened that these Jonas Salk and Albert Sabin, they were fierce personalities, and I mean, like, if you just read about their fighting against they were egoistic clashes, you will be really super amused. Google them out. Anyway, so here what we are looking at right here, just a couple of different things. Polio. Inactivated vaccines, the Jonas Salk vaccine started here. It dropped dramatically in the population, the polio, but could not eradicate it. Once Sabin oral vaccine, which is a live attenuated vaccine, actually came into the market, that actually dropped the cases of polio. And we are going to explain that why it is. So that is the information that you need for the understanding. Measles, the same way, you can just easily see that. And here you can see that 13.8 million people were saved from the measles in the developing countries, say mainly in the developing countries in 2012. So estimated deaths prevented by the measles vaccination, 13.8 million. So it is a kind of a mind-boggling number that what you can see that what large-scale vaccine campaigns can do. All right. You probably know this very, very well by now, is the concept of the herd immunity. Basically, so what happens that when they're in the population, there are multiple people who can get infected, and there are people who can infect, the disease spread. We have already seen that. And now what you have, that you have multiple people, either by natural infection or vaccination. There are not many people. So if there are some people who are even infectious, the disease basically gradually disappear from the population. And this is basically the herd immunity that you know very, very well. So I'm not going to talk about that in detail. So here is one of the things that you take home message from here. This R0, which is pronounced as R0. R0 is basically the ability of one person to infect how many people. That's the R0. So say, for example, on an average, a measles virus infected person can infect 20 people on an average. So the R0 is 20. Say, for an example, for influenza, one person can infect 2 people. So R0 is 2. What is important is that from that number, you can get something. Say, for an example, I'm just thinking about that approximately the maximum number of the SARS-CoV-2 that can transmit from one person to the other, they consider it as approximately, and this number varies, of course, is 4. So what you will see that if you have a number 4, then you can find out really easily that how much of the population need to be vaccinated before it can stop the spread. So the calculation is, see that, or the herd immunity, you can see that the herd immunity is 1 minus 1 over R0. All right, makes sense? So if you just see that this is 1 minus 1/4, 3/4, you're good? That is 75%. So what it means that if 75% of the population in a case where the R0 is equal to 4, if 75% of the population gets vaccinated, apparently the disease should stop. So that's basically the basic calculation. All right, so this is one of the things, it's funny, but that is by natural infection, if the disease or the disease kills a lot of people, basically, say for example SARS-CoV-2, without a vaccination, it is very difficult to achieve. It's a too much cost to pay. Already in the United States, 750,000 plus deaths. So you can just well imagine what it is. So that's what it is. All right, now what we are looking at, we are going to the detail of the vaccine, that how you develop and what are the criteria. One of the most important part of the vaccine, that why it is not easy and it needs years and years of laboratory research in the background, because just getting a response or producing antibody is not enough for a vaccine. So what you have to do, that you have an antigen presenting cells, and they differentiate the CD4 T helper cells into 2 lineages. One is a Th1 that secretes IL-2, 12, IF, and gamma, And another lineage is a Th2, which produces IL-4, 5, 10. So they do different things. The Th2 basically is the antibody response dominates because they just call the B cells. Whereas the Th1 is a cell-mediated immune dominance, and this is a pro-inflammatory response. So the balance cross-regulation is very important. If one antibody is basically one of the virus is basically doing this Th2 response in compared to Th1, it may not be advantageous. So what exactly it is? So think about this way. Say, for example, in acute infection and persistent infection, these cross-regulation is very important. So for example, what do we mean by that? In an acute infection, say for example, this SARS-CoV-2, this Th2 response is actually very necessary. You need a robust antibody response that will basically take care of the virus. That's it. But say for an example, a herpes virus infection, a vaccination against a herpes virus, like think about that, the Shingrix, which is the shingles vaccine, varicella zoster, which is a herpes virus. It's latent somewhere. Where you need that is that you need a stronger Th1X response, which the cell-mediated immunity dominates, because these viruses are not producing outside the cells. So if there are some herpes virus, and this is true for many of the herpes viruses, virus vaccine, where there are cells with the latent infection. They are just viruses inside the cell. And if the virus the cell presents the viral parts, you know that how they display the part over the MHC1, the CD8 cells which are produced, the cell-mediated immunity, they will kill it. The really antibody doesn't have any effect. So it is really complicated that you have to have a proper immune response. Just a response is not enough. So that complicates the vaccine development. So here we were talking about the science and the art of vaccine making, and we'll see that what is the art. There are some empirical factors in there. So what are the different types? We're going to talk about the attenuation, attenuated live vaccines, then inactivated whole virus vaccines. Then fractionation is a non-recombinant purified subunit vaccine. These are usually the proteins which are the capsid protein for naked viruses, or proteins which are on the envelope sticking outside. Those are the proteins which are responsible for this, mainly those. Then you, of course, have the recombinant ones where you can need the replication competent or replication deficient, which is not there. Replication competent or deficient virus vector vaccine. Say, for example, the adenovirus vector vaccine for the SARS-CoV-2, there are several one of them, Johnson & Johnson, AstraZeneca. These are non-replicating adenovirus vaccine, not like this. So there should be another one. The book didn't have those. Then you have a DNA vaccine as well, where you inject the DNA and that will produce basically the mRNAs by the transcription, and then you will get the antibodies against those proteins. And then, of course, the expression of the proteins and the mRNA vaccine, of course, it's in there. So when you have these expression of these cells, what you can do, that you can take a bacterial cell and clone the proteins from the original virus, and the bacteria or even the yeast basically will produce the proteins from that cloned plasmid in the bacterial solution, in the yeast media. And you can just collect them, and many times these basically can have a subunit separate, or they can have a virus particle like vaccine. They can just altogether form a virus-like particle. We are going to see that spontaneously, because we know that, remember that viruses are metastable structure. If you have the capsid protein, they come together automatically. So they can form these VLP, or the virus-like particles. And these subunit vaccines can be packaged into the nanoparticle and can be delivered. Novavax is doing basically this, where they make the spike protein, put it in a lipid nanoparticle, and delivering that. We're going to talk about that. All right, so for some of these classical vaccines, where you have the inactivated, fractionated, some of these vaccines, you in the injectable, which is the injectable vaccine, you have something which we call as adjuvants, but they are not present in the DNA RNA vaccine. The mRNA vaccines, we do not have those. Even with the adenovirus vaccine, you do not have those. Vector vaccine, you do not have that. Mainly, these are present in the inactivated or fractionated vaccines. So what they are, they stimulate the early process and immune response. Basically, they are two things that they just keep the antigen in the place for a little while. It induces a stronger inflammatory response. Your site is painful, red, swollen. And why you do that? Because that will cause a greater immune efficacy. And what are those? Say, for example, aluminum hydroxide, alum, very, very common in HBV. Shingrix contain this monophospholipid A, which is from a bacteria. And what happens that it just engages the TLR4 ligand, and as soon as TLR4 is activated, it just wakes up the immune system to rush to the state and then finds the original antigen, which is the viral vaccine part. Then Ceravix, and this is from the human HPV, alum monophospholipid A. This is squalene in oil water emulsion and innate stimulatory. These are some of the typical adjuvant that you use. And what happens that the one thing is that if you have an adjuvanted vaccine, it's going to hurt a couple of days at your injection site because it is an inflammatory response to that site. But some of the vaccines do not need that, and we're going to talk about those as well. So why inactivated vaccine? This is a still oldest gold standard for sure. You take the virulent parental vaccine, you inactivate by these material. Chemical procedure is formalin, beta-proteolactone, and non-ionic detergents. So basically your goal, the intact viral three-dimensional structure is frozen in time. So all the proteins on the virus are intact as possible so that they can be recognized by the pathogen, recognized by your immune system, and they can have the immune response against that. This means that the cells will take up those, the antigen presenting cells are going to endocytose those, present it to the CD4 helper cell. Then the CD4 cells are going to come over, and then basically that will bring the B cells. They will secrete antibody. Some of them will go into the memory. And the infectivity is eliminated, but antigenicity is not compromised. So here is a fascinating story, and this is just the parts about the story. So you can see that these are the iron lung ventilators, and some of the people who were infected with this poliomyelitis is just polio. They will spend the rest of their life because they cannot breathe by their own in these ventilators. It was like unbelievable that how it was pre-polio vaccine. So this is actually FDR. This is Roosevelt, and a lot of people actually do not know that he actually had polio. So this is a rare picture where he is being helped to get out of the car, and we're going to see that he actually founded this March of the Dimes, and probably do not know that this was developed for a clinical trial for the polio. And you're going to see that when people ask you that, okay, all right, some of the vaccines takes 10 years to develop, how can you develop a vaccine within 6 months or 1 year? Just one thing is the most important one, is money. To run a clinical trial, you're going to see that it is $500 million plus a company has to invest nowadays to run a clinical trial. That's $500 million. How many people or how many companies can come up with that kind of money? So that's what it is. So here is they found that this is the Jonas Salk's inactivated vaccine with the formalin. And in 1954, this National Foundation for Infantile Paralysis sponsored the clinical trial. And this is basically renamed as the March of the Dimes, basically. So 50% protection, results announced in 1955. It was licensed the same day. But this does not stop polio. But here is where there is always a fear with some people. There is an incident called Cutter incident. This was a family-based company who was making this vaccine, and they had a quality control problem. Some of the vaccines were not properly inactivated. That caused, that is kind of 1950s, think about that, 40,000 people actually got symptoms of polio. Out of them, about 200 got paralyzed, about 10 of them died. So these are the things that, although this is 1950, this is 2020, a long time back, but these are some of the things that when there is an inactivated vaccine, people there are still fear those. But there is none of the SARS-CoV-2 vaccine has inactivated vaccines, and chances of them are less. Say, for an example, if this is influenza, really it doesn't matter. Influenza doesn't kill most of the time, like if you're young. But here it was terrible. So the SARS vaccine did not stop the vaccination, and why it is so, the viral route is through the intestine. So what happens that it basically replicates in the mucosal surface before it goes to the lymph node and into the blood. So what happens when the injectable polio vaccine of the Salk, the antibodies, the IgGs, they are in the blood. So what happens, the person who is basically, suppose there is one person, he got infected with the polio. The mucosal surface in the intestine is replicating the virus and the person is shedding the virus. That person will not get infected because once the virus reaches the blood before it's going to the brain, because it's a neurological symptom for the paralysis, the IPV will block that. So this person will not get the disease. But he's shedding the virus because it replicates happily in the intestinal mucosa. So that was the problem. And we're going to see that how the oral vaccine solved this problem. Now let's talk about the influenza virus, and you know that it is a negative segment or segmented RNA. There are 8 of these segments, and these segments recombine in between the strains, and these are some of the challenges for the influenza virus. And there are 3 types, A, B, C types. We're going to see those in there. So here are some of the inactivated influenza vaccine over the period of time. Vaccine are produced a long time in chicken eggs, and in 2019-20, vaccines are all cell cultured. There are no egg derived. Even until 2018-19, there was some egg derived in there. So these are just, you can just easily read that. They are very self-explanatory. So here I was talking about that I just updated this in the morning. The envelope proteins change each year, and there's 2 main envelope proteins. One is an HA, hemagglutinin, and NA, neuraminidase, which is in short H and N. These are the 2 main envelope glycoproteins and the antigens. You will see that it is a little bit different compared to that. So you will see that instead of Singapore, it's probably Cambodia or something that you're going to see. So you're going to see that where they have collected that most of them are the same types, but they're collected and the lineages vary a little bit, and that's every year that how they do this. But this is the basic process that how you develop the influenza vaccine every year. So the WHO basically do this surveillance or the sampling of the virus throughout the year. So by approximately mid of February, the WHO basically selects the most prominent strains that is circulating. And they prepared their reassortants by the end of May and standardized antigen by the May, assigned potency, and by end of August, they are done with the process, and you start from September to vaccinate people. They do this every year. So you can easily see that just within 1 calendar month, it is, say, from here to here, it is 8 months. Every time they have to go through this proper procedure, and they're just developing a vaccine. Of course, you do not need a fresh clinical trial for each one of them because it's already standardized. It's just a different type of antigen. But this is the way that the vaccines are developed every year. All right, so the next part that we are going into is the replication-competent attenuated vaccine. Remember that even the influenza, there are the injectable ones are inactivated vaccine, whereas the ones the flu mist is a live attenuated vaccine. Viral replication occurs, stimulates immune response, infection induces mild, most of the times inapparent disease. There is no disease in there. So what is the difference between that, what we see, that the inactivated vaccine, you need to give the booster and to make the immune response really high. Whereas if you have a real attenuated vaccine, replication competent, just 1 dose can give you a high immune response. That's basically the advantage of that. But of course, there are some disadvantages of that. how you develop this attenuated vaccine. So this is what we talk, that this is almost like a black magic or the art of making the attenuated vaccine. There is no straightforward way. You just do a lot of things and think that the virus will attenuate at some point of time. And this means hundreds and thousands of passages in different cell lines and different animals. This is a very general scheme. Think about that the pathogenic virus is isolated from a patient and grown in a human cell culture. Now you take the virus, and the culture virus is used to infect monkey cells. Monkey cells, it does not replicate, it somehow enters the cell, but it does not replicate properly in the monkey cells. But you just keep trying. And what happens gradually, there's some mutations actually select some of these viruses which make them replicate properly in the monkey cell, not human cell, but monkey cell. Now you isolate them from those monkey cells and try to infect a human cell. They replicate properly because they are adapted into the monkey, and you may get the attenuation strain. It is never that easy. This is just a very general scheme to develop that. So we're going to see some of the details. So this is a flu mist, multivalent, same thing, same strain. So for example, all those 4 strains that we saw that they are in the tetravalent vaccine for this year, they are inactivated when it is injectable. And the flu mist, you have the same strain, but they are in a multivalent inactivated form. These are cold adapted, temperature sensitive, and attenuated usually in a ferret model. All right, so here what we are looking at, this is also the attenuated vaccine, which is the oral polio vaccine, which basically almost eradicated the disease from the planet, except some of the places. So what happens now that as you take the virus orally, it just goes through the natural route and they replicate on the mucosal surface. And now if you remember that, there are these IgAs, which are the secretory antibodies, which are elicit, that gets the response. And right at the mucosal surface, because that's the natural route, the replication is stopped right there. Because this is earlier, only the blood onwards, the protection was in there. But now, as it is going through the mucosal surfaces, all the cells or the immune surveillance cells in the intestine, they bring the T cells, B cells right into the intestine, and the replication doesn't happen in here because the cells are basically, they secrete the IgAs to basically neutralize the virus. And that finally stopped the transmission of polio, and very successful. But there were some drawbacks. We are going to see that. So here, the Albert Sabins actually licensed in 1961. There are 3 strains in there. So we call, in short, we call them as a P1, P2, and P3. So it's a trivalent oral vaccine. And just think about that, how they derived this. 24 passages is monkey, 43 passages is monkey kidney cells. Then non-neurotropism, so neurotropism is gone. 5 passages, kidney cells, monkey kidney cells, 3 plaque purification, and you get the Sabin type 1. 4 plaque purification, 1 passage in chimpanzees, and then you get through. So these are some of the ways that you get these attenuated vaccine strains. But there are some problems also. And this is where the fear comes from. The incidences that we still see with the polio in Pakistan, Afghanistan, and in small parts of Africa, all those are the vaccine strains. What happened? A small amount of these viruses basically were revertants. What it means that after attenuation, once they were injected, they actually reverted back to the proper poliovirus strain and caused polio. So the polio that is still out there, this is due to the vaccine strain, bless you, due to the vaccine strains. And this is where the fear actually comes from, that if it's a live vaccine and if it is something like a polio, which can paralyze you, people are really hesitant to take those vaccines. What if it reverts. So these are some of the logical ways that people just get scared about the vaccines because a lot of people will not know that what you are getting. These are some of the reasons for that. Now we are going to subunit vaccine. So think about that. Subunit vaccine is like under no way that this will cause any problem. There is no live part of the virus, not inactivated, not attenuated. This is just a part of the protein of the virus. Either you have non-recombinant or you produce it from a bacterial cell or an yeast cell and produce the virus-like particle or the subunit vaccine. And antigen usually a capsid or the membrane protein that you can easily see. So here we are just going to describe a couple of them. This is a Shingrix against the varicella zoster, which is a herpes virus or the chickenpox virus. But what happens that it harbors them, you already know, in the peripheral nervous system, in the sensory neurons. And later, after 50 plus, you can get again, the reemergence of the disease from those, and that's what the problem is. So this is basically the recombinant glycoprotein, enveloped glycoprotein. That is basically, so there are many of them, GB through GN, and the GE produced in mammalian in the Chinese hamster ovary cells, secreted and adjuvanted with AS01 and injected. This is your Shingrix. Hepatitis V, this is also a cancer vaccine, remember that. And these are produced in the yeast, recombinant yeast. So you just have these HBsAg, these are the capsid protein. You have the large, medium, and the smalls. And you take these, and then you purify the proteins from the yeast. And what happens that spontaneously, they will form these virus-like particles. They do not have anything other than that. And these are highly immunogenic. You can easily see that. That's assembled into, these are the VLPs or the virus-like particles. They look very different. Doesn't have anything inside the virus. It's just the top. So you can easily see that they're still highly immunogenic. All right, so here is another cancer vaccine, which is a human papillomavirus vaccine, which is an HPV. And this is the vaccine that we should be really being lucky that only basically the developed countries can afford that. Because if you do not have an insurance, that this vaccine still costs about $270 or $300. No developing country can afford this vaccine. So that's one of the things. One of the important thing is that the men and women both should get this vaccine because this is a cancer vaccine and it is not that the disease is restricted to the women as well. So this is a lot of people don't understand. Yeah, so a very, very safe vaccine and a very potent vaccine for sure, HPV vaccine. How it is produced? There are several one of them. So you can see that Merck makes two of them, Gardasil and Gardasil 9, Cervarix, this is from GlaxoSmithKline. And the HPV does have this L1, which is a major capsid protein. And these are produced either in yeast or baculovirus. Baculovirus is an insect virus, and you can make a recombinant baculovirus where in the insect cell culture, you will produce these L1 capsid protein. You purify them, and what happens that it spontaneously forms the pentons, which will make these virus-like particles or the VLPs. See that's the advantage of that. You have spontaneous making of these just by making just one of the capsid protein. The inside is completely empty. Nothing is in there. And very, very powerful vaccine that you can just easily use that. But again, this is a very, very expensive vaccine. All right, so this is something interesting, this is just information, is that the virus-like particle synthesis of HA alone in cells leads to production of immunogenic particle. So that is basically one of the very interesting things. And now, this is the future of the influenza vaccine, the cost. It can be produced in tobacco plants by either transient. Now, they have already produced the transgenic plants. They have put these genes to make these viral HANA proteins in the tobacco plant. And 20,000 doses, 1 square meter plant, tobacco plant, can produce 20,000 doses. Just think about that. You just purify these proteins and it will be like 20 cents per dose. I mean, like it's nothing. So you can see that long-term research can lead it to this very, very easily. But there is still not a universal vaccine. That means there's just one vaccine that will protect against all the influenza. It's still a tetravalent, but there are lots of research that is going on to make a universal vaccine where there is just one vaccine will take care of all the different circulating strain. But still, they are the preclinical, clinical one, those kind of phase trials. All right. After that, we are talking about this Flaviviridi family. And the Flaviviridi viruses, these does have, remember that, on their surface, they have this flat envelope protein. And this is a type 2. Remember that we talked about, say, for an example, the HA1 or the H1 of the influenza, or say the spike protein of the SARS-CoV or any of the SARS, they stick out. These are the type 1s. They have a stalk. On top of them, you have a globular head. Whereas these, they lie next to each other like a beta sheet, and then they open up in a different way. So they are just a kind of a flatter protein. They do not stick out. What are the very common flaviviruses? Yellow fever, dengue, Zika. These are your most common ones, and many of them are mosquito transmitted. And the yellow fever vaccine was produced in 1938 by 176 passages of this in the SCB strain in the chick embryo. So, I mean, like, just imagine 176 passages. It's like unbelievable. And large doses have been distributed, safe, effective. Now, the yellow fever vaccine is a travel vaccine for the United States. If you just go to certain part of the world, you need to take it. Otherwise, you do not have to. On the other hand, the dengue, we are going to talk about that. There are some vaccines for there. Zika, still clinical trial. So, this is how the dengue vaccine was built. One of them in this process, where they take the backbone of the yellow fever vaccine, yellow fever virus. So here you can see that they took this one, and this was basically the backbone of the yellow fever. And right here is the envelope protein, and they changed this and the membrane protein. See that this is a membrane protein, envelope protein right here. These are the two on the top, and they replaced this because remember that they are all Flaviviridi family, so the dengue virus will also have this membrane protein and the envelope protein. They replaced the backbone of the yellow fever with this. Now you have this chimeric virus, and that's also attenuated, and that was the vaccine. There were some problems with this. So this was the EPRM of dengue with the YF70D backbone, which is the yellow fever vaccine. And this is licensed in Mexico, Brazil, and the Philippines. But this doesn't do anything against the dengue virus type 2. It is not licensed in the US and many other countries because there is a problem. What happened that sometimes it can lead the worst disease into 2 to 9 years. And although it is licensed in the Philippines, there were 800 children after getting the Denvaxia actually got seriously ill after the vaccine. So you can easily see that even these type of vaccines do elicit fear to the population that something bad could happen. So this is not licensed in this country. So now this is what the best dengue vaccine is. This is developed in NIH, in the National Institute of Health. It's called a TV003, and another one is a TV005. What they did that they took the flavivirus RNA. Remember that all these Flaviviridi, these are plus sense RNA strand virus, single strand plus strand RNA virus. SARS-CoV-2 is the same, a different family, but they're the same, plus strand RNA virus, 5' to 3'. What they did is a 3' UTR untranslated region. They found that they deleted 30 nucleotides, and that makes the virus attenuated. Remember that we already talked about that there are some of the viruses where the untranslated region are the virulence factors. They interact with the microRNAs, silencing RNAs, and they are essential sometimes. And this is, they already made a tetravalent attenuated dengue virus. So there are 4 different types of dengue, and it protects, again, all of them. And one dose 100% protection. I mean, like, you do not get these type of things. So you can just easily imagine that how effective that vaccine is. So this is an amazing vaccine from NIH, licensed from NIH, I mean. Now, this is the Zika, what it does. See that in adults, you can have in this Guillain-Barré syndrome, myelitis, encephalopathy, many of these. Whereas in the pregnant women, if they get these, sometimes the infants will be born with a microcephaly. You can see with the microcephaly, with severe and moderate microcephaly. So these are some of the disease. And that is what it is. So they are trying to get these vaccines, and how they are basically trying to do that they are still under the clinical trial. Nothing has been licensed yet. And the thing is that we are going to explain that why it is not so, because the thing is that the incidences of the Zika is not many. So for the vaccine trial, think about that. You have to inject the vaccine for the clinical trial to 30, 40, 50,000 people. Where you are going to get this many volunteers to have the vaccine and exposed to the Zika virus? It's very difficult to get that number of population, and of course, along with that, the money. That was one of the reasons that Ebola virus vaccine took 10 years to develop. It is not a problem of the wealthy countries or the Western world. It happens in the pockets of Africa. Nobody wanted to invest $500 million to develop an Ebola vaccine, because they will probably never get back that money. They just spend that, and then in Africa, they have to give that vaccine basically for free. And these are some of the impediments that the companies actually have, why it takes so much longer time to develop. So now let's talk about the replication competent recombinant virus vaccine. So they cloned these virulent parental virus. They take the genes and take the specific genes for, mainly if it's an envelope virus, the envelope glycoproteins which are sticking out, or the capsid protein if it is a naked virus without an envelope. They clone them, and then what they do that they make an infectious clone. And this is a replication competent virus where it will display these envelope or the capsid protein on their surface. So let's take a look at how this is possible, how you can do that. So this is the one which is in a trial. So here what you are trying to do, that just understand that how this is done, this is what is called a pseudo-typed virus vaccine, vaccine vector. So here, the vector is a vesicular stomatitis virus. Now remember that this is an animal virus. This is not a human pathogen. So it doesn't cause any disease in the human being. But it can replicate or enter into the human cell as well. It contains a surface glycoprotein, like G. And right here, this is the G, this is the glycoprotein of the VSV. Right here, this is the gene. Now you take the Zika virus, remember that it is also the flaviviridi, and you have the envelope protein right here on the surface. You take the envelope protein and replace this G glycoprotein of the VSV with this E glycoprotein. Now you package the virus basically in the cell culture, because now what will happen, you will have a recombinant VSV where instead of this own glycoprotein, it will have this envelope protein on the surface of that. You just inject that, and this is your recombinant VSV vaccine. Your Ebola vaccine is exactly the same. It is based on a VSV, vesicular stomatitis virus, and the Ebola virus glycoprotein from the surface is on it, which is already approved and licensed. And the trial for the Zika is with this. This is called a pseudo-typed virus, whereas that it is a vesicular stomatitis virus with an envelope protein from a different virus. That's why you call it pseudo-typed virus. All right, so finally, for the COVID-19 vaccines, and there are two that I just did not highlight, is that there is a DNA vaccine that is being developed in India. This is by the Zydus Cadila. We call the ZyCoV-D. They are under clinical trials, so that's why I did not highlight yet. They are, and another one is the Novavax. What they did that this Novavax basically produced this spike glycoprotein, which is on the surface, you all know that. They just produced that large amount, the subunit, and then package them in the liposome to deliver. And they are also in the phase 3 clinical trial, not in the regular population. Now what you have, mRNA, you have the Moderna, Pfizer, and then you have right here in the United States, the most common one is the Johnson & Johnson. Now, the Janssen is the company which developed that. This is basically a Belgian-based company owned by Johnson & Johnson. That's why you will see both of these names. And a lot of people ask me, is like, somebody says it's a Janssen's vaccines, Johnson & Johnson vaccine, what's going on? So Janssen is a smaller biotech company which is owned by Johnson & Johnson. On the same way that BioNTech is a German company which basically developed the vaccine and partnered with Pfizer because they don't have the money to run the trial, basically. And these are some of the facts of this. So Moderna is also a small biotechnology company which did the vaccine. But from the government, from federal money, from the BARDA, which is the Biomedical Advanced Research and Development, and they got how much? 483 million, close to $500 million, something, 480 or something. And now after that, so think about that, close to $500 million to run the clinical trial. Pfizer, on the other hand, is a giant company. They sponsored their own clinical trial. And where these money comes from, the companies cannot just do that. So that's where it is. Johnson & Johnson also received $450 million from the government. So Johnson & Johnson is a fairly big company. But even they couldn't sponsor the clinical trials that is involved or the cost involved in there. A couple of other vaccines, adenovirus VAX. This is an Oxford AstraZeneca vaccine licensed in many countries. CanSino, this is from China. Sputnik 5, these are in Russia. These are all basically now either completely licensed, approved, or used in emergency phases. There are two of them which are inactivated as well. So one of them is the Bharat Biotech Covaxin. This is an inactivated vaccine. This is now approved and also WHO approved. Some of these vaccines are licensed and approved in some of the countries, in not all the countries, but some of them are also approved by the WHO. And the CoronaVac is a Sinovac. This is a Canadian-Chinese company and being developed in China. That is also available in the market. So this is probably the most common COVID-19 vaccine that you have. But in the United States at this point, that if you're just trying to get something, you will either get a Moderna or Pfizer or the Johnson & Johnson, which is adenovirus-based, where these are the mRNA-based. So let's first talk about that, how you make the adenovirus-based vaccine, because you know the virology of this. So what you do, that you take a parental adenovirus, and there is an E1 gene. You already know that what E1 does. E1 is the transcription factor essential for the transcription of the late genes, E1. They replace that E1 because it's a transcription factor, and in the place, using a shuttle vector, you put right here on the E1, you put the spike protein, basically, with a secretory signal so that it goes to the membrane. Remember that you have to put a signal peptide in there and a secretory sequence so that it goes into the membrane. Now what you do, that you basically deleted that, you package that, and this is your recombinant adenoviral vector with your gene, and now you recover this adenoviral vector and infect or transfect the cell line, which, this is the most important part, the cell line should express the E1. Otherwise, the virus is not going to replicate. So remember that. Virus E1 protein deleted, which is a transcription factor essential for the viral replication. That is substituted by the spike protein. Now you choose a cell line, and there are some of the cell lines which already have these transcription factors in them. So one of them is a very common, is HEK293, which is it's all in your notes. It's a human embryonic kidney cell line 293 that expresses E1 constitutively. So if you just put this vector of the adenovirus, which contains everything except the E1, and here it is replaced by the spike protein. It will replicate in the cell line, and as you have a secretory signal of the spike protein, now you have a funny virus, which is an adenovirus, and the spike protein is sticking out of it. You just purify that virus, and that's your vaccine. So now what you do that you inject that, you have the spike protein on the surface, you're going to the human being, it's just going to deliver the antigen on its surface to be recognized. It's not going to replicate because there is no E1 gene, and the human cells doesn't have that. Normal human cells doesn't have any E1 gene. So it's a replication incompetent virus just for delivering the antigen. That this is the technology that what you use. And depending upon that, the AstraZeneca used this. See, for example, if you see that why they call the CH had ox in there, because they just uses the chimpanzee adenovirus, whereas adenovirus type 5 was used, which is a human adenovirus. So depending upon that, there are many different types of adenovirus vectors were used. This is also the same way. This is just a little bit different. I'm not going to go into the detail of that. That you have a shuttle vector, and here you have an adenovirus molecular plasmid. You just cut this basically. So the previous one, this was by the recombination, homologous recombination of introducing the spike protein. Here it is just cut and paste like regular cloning. And here you have the recombinant adenoviral molecular plasmid, which contains all the genes for making the adenovirus, except the E1, which is replaced by your spike protein. And you just put them in the 293, HEK293, or any suitable cell which is expressing E1. The virus will basically produced in that cell. You just basically harvest those cells and purify the virus. That's it. So this is your adenovirus vector vaccine. Now is basically the mRNA vaccines. And I'll just go through that. Suppose you are just trying to make an mRNA vaccine in the laboratory. And technically, you can. Whether it will be how much efficacy it will have, that's a different thing. But literally, these reagents you will be able to make these vaccines at a laboratory, at a molecular biology laboratory, anywhere, like here in a Clemson laboratory who works with a laboratory. So this is basically an in vitro transcription. This is being done from 1970s. I mean, like I used to do that every day in 2000s in there for a different vesicular stomatitis virus, but in vitro transcription the same way. And this is a protocol from New England Biolabs. If you're just working in a lab, you'll see that this is a company which supplies all the reagents for that. So basically what you do, you have the sequence of the spike protein. And then from the sequence of the spike protein, you have that, and you can just isolate the viral RNA or synthesize it chemically. And then you can do a PCR amplification of that. Or you can clone the spike protein gene into a bacteria, from where you can amplify that. And then you purify the fragment. This is your DNA fragment. Remember that double-stranded DNA fragment. What you do usually, the double-stranded DNA fragment, in front of the DNA fragment where the sequence or your ATG, the first start codon starts, before that you need a promoter. Remember that, you need a promoter from the RNA polymerase. And usually what you use, you use a T3, T7, or SP6 promoter. These are all bacteriophage promoters. Bacteriophage promoters, and you just put them in there. So now you have the double-stranded DNA. You have one of the promoters and the spike protein. Now, in a test tube, what you do that you need to put these RNase inhibitor, pyrophosphatases, DNases, nucleotide triphosphatase, and along with that, you put, if you have a T3 promoter, you put a T3 RNA polymerase. You have a T7 promoter, you put a T7 RNA polymerase or SP6 RNA polymerase, just one of them. These are like all the kit all together, but that's the manual process that what you do. So these T3, T7, SP6 RNA polymerases, these are bacteriophage RNA polymerases, which basically is purified, then cloned in bacteria yeast, and you can just get, if not liters, of these from these biotechnology companies. They are produced, and anybody can buy this. So see a 200 microliter of T3, T7, or SP6 will cost you about 44 or 45 bucks, 200 microliter. And out of that, you can just make almost 1 milligram of RNA. I'm like, you do not have to remember the numbers, but I'm just saying that just think about that, how easy it is to do that. So now what you have, that from the double-stranded DNA template, you have the RNA. This mRNA is not capped, not polyadenylated, contaminated with the DNA. So that's why you have a DNase. And also remember that as it is being synthesized, you have the NTPs. When each nucleotide is getting in, it was also going to produce these pyrophosphatase, PPIs, 2 PPIs, because 2 phosphate goes out from the triphosphate, 1 phosphate stays in the RNA. Those are going to be degraded by the pyrophosphatase. And DNase 1 is going to degrade the template. So everything else other than the RNA is degraded. You purify this. Now you go to the next step. In the next step, you can use a vaccinia virus capping system, and it contains all the capping proteins, 3 usually. So you have in the right here, you will see that I've detailed that. You have the guanyl transferase, you have an N7 methyl transferase. All of these are together, and also along with that, you need to remember that what you have to do, that you have to put a cap at the 5' end of the RNA. So you have to remove that 5' phosphate from there to put another one. So for that reason, you need a phosphatase in there, and those are the things. So phosphatase and the N7 methyl transferase, guanyl transferase, and you add GTP in there. And for the methyl donor, you need to add SAM, or the S-adenosyl methionine. This is your reaction mixture along with the buffer. You put that there, the cap is now put on the RNA that you built in the previous step. Also, what you can do, that you can add this 2' O methyl transferases, not necessary, but this can just make the cap 2. If you remember that, the caps have a cap 0 or 1 structure and a cap 2 structure. So you have a GTP, and then usually the next A's or the adenines also sometimes have these additional methyl residues, which is called a 2' O methylation. And this enzyme, in addition to that, will add those. So now you have the full, complete, mature cap at the 5' end of the RNA. You still don't have the poly A tail. You add the E. coli polypolymerase. That's it. That next step, you purify this E. coli polypolymerase, it will do that. So this is manually, but see that this is your HiScribe T7 ARCA-mRNA synthesis kit with tailing. All you do is you have the DNA made, you just add these in a single mixture tube. It's going to do it all for you. Just purify your mRNA. That's it. So it is really that easy. So not anymore. Still, when we used to do in early 2000, we used to do it completely manually, this way, not this way. Anyway, so very, very effective, and you can just look at the, you can just do this spin column chromatography. You can just purify them. Of course, in the industry, you have to do it in a large scale. Next is that you have to package them for the delivery. So what you do, that you just make liposomes out of the membrane lipid. These are the phospholipids along with the cholesterols. And then you package these mRNAs into them, which will basically deliver them into and package them in that. So here I'm not going into the detail of that. And here, this is a funny video. A couple of kids, I think that they're English, so they have an English accent. They were making liposomes, and it's just kind of funny that they were packaging methylene blue inside those liposomes and just showing that. So basically, you take the lipids and mix it with ether, chloroform, some kind of a very highly volatile liquid, and rotate them on a rotaevaporator flask. So the flask is coated with a lipid. Then you add the mRNA in that, you just add the buffer. As soon as you add aqueous buffer in there, the lipids are going to just come off the surface of that glass and just going to enclose the lipid, and you have these lipid droplets. That's it. And that will be your liposomes. And this is from the inserts from the CDC USA. See right here, these are the lipids that you use. These are the membrane lipids, and these are for the liposomes. So you have the mRNA, in the vaccine, and mainly these are the stabilizers, which will increase the shelf life. There is no adjuvants in those. Now you deliver the lipids into the cell. So now what happens that it either goes to a muscle cell, some will leak into the intercellular space in the interstitial fluid. So let's see that if it gets into a muscle cell, what's going to happen. Once it goes into the muscle cell, the mRNAs are released. The ribosome will read them, will make the spike protein. And the cell will spit some of them out because there is a soluble secretory sequence in there. Some of them are going to capture because this is a foreign protein. Cells have it's like, okay, all right, this doesn't have any usage. Why is it sitting in there? And they will degrade some of them. And as you know, that as proteasome degrading this protein, it will just show up on the surface of the cell by that MHC1 complex right here. CD8 cells not yet coming, but it's just showing. On the other hand, what can happen that other than this MHC1 pathway, think about that these spike protein which are spilled out of some of these cells, or some of these liposomes can be endocytosed by a dendritic cell or a macrophage, which are doing immunosurveillance in your muscle. They are all over the connective tissue. They can pick up by the endocytosis, they will degrade it, put it on an MHC class 2. As soon as they put it on an MHC class 2, they will show it on the surface, and these will now migrate to the closest lymph node and present this to the CD4 helper T cells. So CD4 T helper cells now is going to do the Th1 response first. By the Th1 response, it's going to tell the CD8 cytotoxic T cell that go look out for this antigen who is presenting this. Now you have a muscle cell that's showing this, is going to kill the muscle cell. So that's your pain, that some of the muscle cells will be killed. And even that in between the muscles, you will have some fibroblasts in the membranous fibroblast. Some of the fibroblasts can also get killed. And after that, the CD4-Th2 response, that will call the B cells to secrete the antibodies. And now basically what happens as the muscle cell basically is killed and lysed, all the spike proteins that were inside will come out in the interstitial fluid. They will be picked up by the B cells, and the T cells will recognize them. You will have a robust immunity, and they will basically get over. The last things that I'm just just showing that this is a conventional mRNA. What is in the horizon that you produce a self-amplifying mRNA where you put an RNA-dependent RNA polymerase along with the mRNA, which can amplify them in the target cell. So this is in the horizon, which is either self-amplifying in a cis or in a trans. So think about that instead of 10, 25, 50 microgram of RNA, you can probably just inject 1-2 microgram RNA of the self-amplifying RNA, and that's going to give the same kind of immune response. You can make it against all the different strains of the viruses already in preclinical trial. All right, thanks for your patience though. Long one, but it was worth it. All right, have a good weekend. We'll just open up this. I will keep the quiz open until Monday, the same way what I'm doing, just one extra day. So hopefully we have a Wake Forest this Saturday. Good luck. You better not be happy on Saturday, I guess.